We have therefore made the assessment that COVID-19 can be characterized as a pandemic.
विगतको लगायत गरेर 3 वटा हालसम्म चाहिँ पोजिटिभ देखिएको अवस्था छ।
114 पुगेको छ।
देशभर संक्रमित 1846 पुगेका छन्।
15259
21000 नाघेको छ।
44236 90000
1,86,000 पुगेको छ।
On 29th January 2020, the first case of COVID-19 was detected. It was then characterized in the laboratory of the Center for Molecular Dynamics Nepal, working closely with the Government Disease Control Unit. And since then, we have been working relentlessly to prepare for COVID-19 in the country. But how did we even get here? To understand that, let us rewind the clocks a little bit. We have been conducting Nepal's longest running virus surveillance program from 2012 to 2019 called Predict Nepal in collaboration with University of California, Davis. This project has collected human and animal samples from both rural and urban wildlife interfaces. This then helped to detect, track, and better understand the emergence of new pathogens from wild and domesticated animals that could pose a threat to human health. Altogether, 26,217 samples were collected from 3,293 animals and 2,048 humans during 8 years of the project. These samples were screened for 5 viral families, including corona. Kathmandu's unplanned informal settlements along the rivers were targeted for studies in urban wildlife interface. These 4 sites were selected because they were at a risk of wildlife contact and had potential for zoonotic virus transmission and spread. There we sampled both wild and domesticated animals and conducted behavioral risk surveys. In the Jadibuti slum area, we also detected the coronavirus in ducks, humans, and rodents. For rural wildlife interface, we traveled 170 kilometers south of Kathmandu to Silinge, a small village in Makwanpur district. The village is mostly populated by the Tsepan community, one of the most disadvantaged indigenous communities in Nepal. They also traditionally have fruit bats as part of their diet. We have been collecting samples from humans, domesticated livestock, and also wild animals like monkeys and bats. Here we have detected Hendra viruses. During the project period, 19 unique viruses were detected in animals and people. In the first phase, we sampled wildlife in urban areas of Kathmandu and detected corona, paramyxo, hanta and rhabdoviruses in rodents and shrews, Henipa and retroviruses in monkeys and coronaviruses in bats. In the second phase, we sampled animals and people in Kathmandu and Silinge. We detected 3 paramyxoviruses, 2 influenza, and dengue virus. This brings us back to now. Learning from the past 8 years of working with emerging disease preparedness through the Predict project and utilizing all the tools and techniques we developed, we are now working with Madhyapur Thimi Municipality Office to surveil, screen, and in turn contain COVID-19 in the area. Along with a molecular diagnostic laboratory at the Nepal-Korea Friendship Municipality Hospital, a systematic and secure geoinformation system-based database has also been created with information of high-risk residents. This has helped the municipality to conduct hospital and community-based testing and surveillance of COVID-19, including any contact tracing if necessary to uncover possible community transmission and provide a complete service to deal with potential COVID-19 case in the area. Madhyapur Thimi is relatively smaller compared to other metropolises in the valley. To tackle this problem, we have been using innovative tools for environmental surveillance using sewage samples. In June 2020, we successfully detected SARS-CoV-2 from sewage samples from 2 of the 4 sites used for the study. CMDN's past work in other environmental disease surveillance projects involved mapping out of manholes, their outlets and respective catchment areas covered by those outlets in few heavily urbanized regions of Kathmandu Valley. The fact that we were able to swiftly diagnose SARS-2 and COVID-19 in Nepal is a testament to the importance of disease prevention efforts like PREDICT. We cannot ignore the increasing COVID-19 cases in the country, but our efforts in building capacities to surveil, detect, and characterize pathogens in high-risk areas has helped us understand disease outbreaks and epidemics. And now, we're definitely better prepared.